Now, because of all what I told you before, I want to just give you 2 slides of the stuff that we are doing. We believe that we need to repair the brain, and we should do it because is a need for patients that are with multiple sclerosis that is advanced. And therefore, what we would like to do is to engineer the stem cells so that they can survive the inflammatory environment of the brain. Now, there's no point in putting a stem cell into an environment where we know already the stem cells are dying or they cannot function. So we need to make cells that are immune to the pathological process and that they are capable of differentiating in the brain, so we tell the cell what to do. And the other thing that we are doing is to design new molecules that are neuroprotective. So molecules that are capable of fighting the inflammation or giving to the cells signals that will allow them to differentiate to what they need to do. This is a work by Dr. Alex Arenkov, and he has a poster outside. And what we are doing seems simple, and it's quite simple, really. What we do is we engineer a receptor on the cell surface of the cell. This is the cell surface of the cell. And this receptor is part of an antibody that recognizes a protein that is in the brain. So first of all, now we are making the cell recognize something that is there locally. But inside the cell, we fuse to this different signaling domains of, you have heard today that interferon induces genes. All these things induces stem cells and oligodendrocytes either to differentiate or to survive. And we have now engineering stem cells that are capable of surviving the presence of pro-inflammatory cytokines like TNF, interferon gamma, that we know are cytotoxic for those cells. So these cells are capable now, engineered cells, to survive that inflammatory milieu. And the other thing that we are doing is engineering a new type of products that can only be activated at the site of disease. So things that will not have side effects. We have heard of side effects today quite a lot. These are biologicals of the type of interferon, and the idea is that we fuse already a protein that is produced by our body that is derived from something else called transforming growth factor beta that makes a shell around a drug or therapeutic component and has a release mechanism that is mainly found at sites of inflammation. So when this product is around the body, nothing happens because it's not being released. When it gets to a site of inflammation, there is a release mechanism that cleaves the therapeutic component, gets released, and then can have an effect in the tissue that you want to treat. And we have put all sorts of things in there, among them interferon, and we are putting also some other neurotropic agents like insulin growth factor and erythropoietin, and we are testing these things in the animal models with the doctor of the mouse that you heard before. And I will stop here, and if you have questions, please. Thank you.